Ashok Singh, Vice Chancellor and Director, ICAR, IARI, New Delhi. I also welcome Dr. Rashmi Agarwal, Dean and Joint Director, Education, Head of Plant Pathology, IARI, Dr. Robin Gugai, Secretary, IPS, Dr. T.K. Bag, President, IPS Delhi Zone and Organizing Secretary, Dr. Amrita Das, Zonal Councillor, IPS Delhi Zone and Co-Organizing Secretary. So now I request Dr. Rashmi Agarwal, Dean and Joint Director, Education and Head Division of Plant Pathology, IARI, for the valedictory address. Please, ma'am.
Thank you, Ritika. Sir, Director Sir has joined, no?
Yes.
Good afternoon, everyone. Chief guest of today's function, Director IRI, Dr. A.K. Singh, who is an eminent rice breeder, internationally acclaimed. Dr. T.K. Bag, who is President, IPS Zonal Delhi Chapter. Councillor Dr. Amrita Das, Faculty of Division of Plant Pathology, Secretary, IPS Dr. Robin Gogoi, Joint Secretary, and Treasurer Dr. Malkhan, the faculty from Division of Plant Pathology, and also faculty from Division of Quarantine from NBPGR, and many other members of Indian Phytopathological Society, research scholars, students, ladies and gentlemen. It's really a very proud moment and pleasure for me to welcome our honorable chief guest, Dr. A.K. Singh, Director IRI. Sir, we are very thankful to you and warmly welcome you, sir, to this evening in spite of your very, very busy schedule. I know that you just left for lunch and immediately you came back, and you are hard pressed with a lot of other assignments. Immediately after this also there is another assignment. Sir, we are so thankful, and by your gracious presence and also with your motivational words, because this zonal symposium was organized, sir, for 2 days on an important topic, that plant diseases impacts on food security. And ensuring the food security along with nutritional security is one of the main goals of UN's, that zero hunger, and we have to remove the malnutrition and hunger among the population. And sir, the 2 days deliberation was very nicely conducted by Zonal President Dr. Tushar Kanti Bag, and that report will be presented soon after my this just formal brief welcome address. So I would like to welcome all the faculty members, all distinguished speakers, invited speakers, and there were some keynote addresses also during these deliberations. So it's my really a very warm welcome to all of you on this valedictory, the conclusion day of 2 days symposium. And at the end of this symposium, I'm very sure that, sir, I would like to inform you that many of the research scholars and students, they gave poster presentations, and the poster presentations were really, some of them were excellent, sir. Very nicely, they young students of our division, MSc and PhD students, and also some scholars, they presented very well the posters. And also in addition to that, there were some invited lectures and also keynotes lectures were there. So I warmly welcome each one of you on this valedictory function today, this afternoon, sir. With these words, I'll stop here because the report has to be read, and also subsequently, sir, we will hear you, sir, for your motivational guidelines. Yes, to organizers, Ritika.
Yes, ma'am. Thank you, ma'am. So now I request our honorable chief guest, Dr. Ashok K. Singh, Vice Chancellor, Director, and ICARI.
It has to be Ritika.
Yes, ma'am.
Before we invite sir, we have to have a report on the 2 days deliberation.
Yes, President.
Yes, Dr. Bag.
Yes, Amrita, I think you give wrong information. Okay, I am starting.
Otherwise, I couldn't.
Good afternoon, everybody. Respected Chief Guest, Dr. A.K. Singh, and Director and Vice Chancellor, Indian Agriculture Research Institute, and Dr. Reshmi Agarwal, Dean and Joint Director, IRI, and Head Division of Plant Pathology, Dr. V.K. Baranwal, Professor, Division of Plant Pathology, Dr. Robin Gogoi, Secretary, Indian Phytopathological Society, and Dr. Amrita Das, General Counsellor, Dr. Sharan, Dr. Poon, Dr. Pant, and many other participating in this program. Now it is the end of the program. Now I have to give a little bit report. In this report, I want to mention that in technical session there is two keynote lecture, and Dr. B.P. Singh, former director CPRI Simla, delivered a keynote lecture on managing impact of lead blight disease on potato production in India. Dr. Singh said that lead blight pathogen Phytophthora infestans and potato has been studied in detail during last 72 years, and robust management options have been developed and put in practice to manage the disease within the economic threshold level. However, he emphasized to study the current status of lead blight disease in India on potato. Second keynote lecture was by Dr. S.C. Bharadwaj, Head and PS Regional Station, Indian Institute of Wheat and Barley Research, Flowerdale. He gave a nice keynote lecture on a century of wheat rust research in India from race to genomes and genomics. He said during the last 35 years, more than 35 pathotype of Puccinia species on wheat have been identified in initial stages along with rust-resistant sources. In discussion, it came to know that three important rust races were sequenced in India. They are different, but where are their similarity, it is convergent, and differences, divergent in sequences are need to be further studied. And in invited lecture, while Dr. Celia Chalam presenting the role of diagnostic and quarantine regulation in crop biosecurity, she mentioned that because of strict implementation of post-entry quarantine, many plant pests and diseases have been prevented to enter in India, but these pests and diseases are present in other countries. There is limitation for identification due to unavailability of diagnostic kit of specific disease of viral nature. Therefore, there is a need for national plant pest diagnostic network. There is also need for proper regulation on new pests and diseases. Separate budget is required for pest and disease diagnostics. Dr. Ramesh said that bacterial wilt of crop is very important throughout the world, but no control measure was found suitable for its management. So he informed that at present classification of Pseudomonas rhodiosus has changed. According to him, Pseudomonas rhodiosus has categorized into three categories: Pseudomonas pseudoceroniacearum, which contains phylotype 1 and 3, and Pseudomonas CGG, which contains phylotype 4, and Pseudomonas solanacearum, which was original name earlier, that practically belongs to phylotype 2, mostly from potato origin. And rest is technical session is Professor M.J. Narasimhan Merit Academic Award. In this award, 5 contestants was there from different discipline. Among 5, all of them presented well, and one was there IPS travel grant. One participant was there, but finally he cannot present here because of some difficulties. And in poster session, there is 16 poster. And theme area, taxonomy and diversity of plant pathogen, there was 3 poster. And theme area, host microbe interaction, there was 3 poster. And theme area, plant disease management through bioagent, chemical, and resistant breeding, there was 5 posters. And theme area, plant pathogen diagnostic, 3 poster, and pathogenomics is 3 posters. This is all about the presentation and recommendation. Sir, may I present the result of this poster and this academic award now, madam? Along with this.
Yes, Dr. Bagh, you can give the complete report and then we will invite our chief guest.
Okay. And Now it is coming to the result of poster session. The session is taxonomy and diversity of plant pathogen. Their first is Asmit Singh, and second is Nadia Manjar. And second thing is host microbe interaction. Their first is Firoz Mandal, and second is Amit Kumar Keswani. And the theme plant disease management through bioagent, chemicals and resistant breeding. Their first is Sunil Jadhav, second is Gaurav Kumar Jadhav, and third is Shashi Pandey. Plant pathogen diagnostic in this theme, first is Srinath YS, second is Soibba Singh, third is Surabhi Mitra. And theme area pathogenomics, first is Arthi Nikekani, and second is Deepak Kumar Mohanta, third is Sweta Agarwal.
So,
And in result of Professor M.J. Narasinghan Academic Merit Award, there are 5 candidates, and first position goes to Kuleshwar Prasad Sahu. He got highest mark, and second is Firoz Mandal, the second highest. And according to the rule of Indian Phytology Society, only 2 will go to the final presentation when it will be organized in Delhi. These 2 are qualified. First is Kuleshwar Prasad Sahu and second is Firoz Mandal. With this, I end this presentation of this.
so congratulations to all the award winners
Okay.
and those who selected for MJ Narsimhan contest to be organized at national meet. So we clap.
Thank you, ma'am. Thank
Yes,
you so
yes.
much.
Thank you, sir. So now I kindly request our Honorable Chief Guest, Dr. A.K. Singh, Vice Chancellor and Director, ICAR, IARI, New Delhi, to kindly address the gathering. Please, sir.
Thank you very much, Dr. Rasmi, Dr. Bernwal, Dr. Bagh, all the distinguished colleagues from the Division of Plant Pathology, students, participants, for inviting me to the valedictory function of this important symposium on new emerging plant diseases, which is an extremely important subject area. In past, we have seen the impact of the new diseases, particularly, for example, wheat blast has been a devastating kind of situation in Bengal, and enough preparedness, timely preparedness by global community has helped in containing wheat blast spread in India. Collaborating with the multi-institutional network, CG centers, having locations for screening in the areas which are classified as hotspot is what has really saved us. We all know the history of rust in this country, and the very Division of Plant Pathology played a very important role in blocking the Puccinia path by suggesting the deployment of resistant varieties in the central India. But for that kind of study on epidemiology and corrective measures, it would have been a difficult thing to do. Therefore, this particular symposium, which you have organized and very important points have been discussed, is extremely important. We also know that the United Nations has declared the year 2020 as the International Year of Plant Health. And it is estimated that by 2050, we need 60% additional food grain production for our estimated population of about 10 billion people on the earth. Now, this increase in production, along with reduction in food loss due to pest and pathogen and food waste, will be needed to meet this demand. Otherwise, it is not possible. How much we lose after production at a significant cost is something which is a matter of worry. Particularly if you see, for example, in case of cereals, we have estimates that 10% of the total production is lost during post-harvest storage, handling, etc. And primarily because of the stored grain pathogen infection, aflatoxin, and things of that kind. 10% of the total cereal production, total food grain production, which is 309 million tons, is almost 30 million tons. So you can see 30 million tons we are losing because we are not able to store it properly. And this 30 million tons of production comes at a huge cost of environment, fertilizer, water, labor, everything we spend and we produce, but we are not able to save these. So that's a real matter of worry. Situation is much more worse in case of perishable, particularly if you see fresh fruits and vegetables, we lose almost 40%. The total production is 329 million tons, and out of that 40% is roughly 120 million tons of fresh fruits and vegetables are lost because of poor storage, because of disease infection in post-harvest situation. This is a huge loss, you can imagine, and the cost at which it is produced, this is several times more than the total production of food grains, fresh fruits and vegetables in many countries. And therefore, not only management of plant diseases during the crop growth phase, but also post-harvest management is an extremely important area that we have to focus on. The losses that have been globally estimated is about 21% in case of wheat, 30% in case of rice, 22% in maize, 17% in potato, and 21% in case of soybean. So this is a huge loss, which is in true sense a matter of concern. Now, what are emerging plant diseases? Which diseases can be classified as emerging plant diseases? Basically, there are a few criteria to call them as emerging. The diseases that have increased in their incidence, geographical area, and host range in recent past have changed their pathogenesis. This is another important criteria. Have newly evolved and have been discovered or newly recognized are the diseases that come in the category of new plant diseases and emerging plant diseases. And these are a matter of global concern and cause a heavy toll on food production and social and political stability of nation. You all know, I need not to say to this gathering, that a classic example is that 19th century Irish potato famine. In 1845, Phytophthora infestans destroyed the potato crop, and in subsequent year it led to Irish famine with over 2 million deaths and a mass migration of people from Ireland to other places. Likewise, the conflict, poverty, and British rule during World War II led to Bengal famine, which primarily was attributed to the brown spot disease of rice. This was the time when there was a Japanese occupation in Myanmar, and you know that we used to import a lot of rice from Myanmar. Burmese rice used to come in India, and because of this Japanese invasion there, the rice from Myanmar could not come to India, and Indian crop was destroyed in that region because of brown spot disease. And that led to millions of people die because of the, this what we call as Bengal famine. Then, recently we have had the coffee rust outbreak in Central America, which provides yet another example of displacement of people due to an emerging endemic plant diseases and climate change. What is interesting here to see is that, diseases, they move from one country to other country, from one boundary to other boundary. They do not recognize the political borders, but humans do. We do recognize, which is a matter of concern. We do recognize, and many times we become restrictive in our approach. There is some knowledge, information available about a particular pathogen in particular continent or country, and people do not want to share. not willing to share information on plant diseases outbreak. Better communication is required. Likewise, the new trade relationship likely to increase migration of plant diseases. Now you see there is a trend that you don't produce everything that you need. Of course, we are talking, in India, our Honorable Prime Minister has given call to become Atmanirbhar, and Atmanirbhar means that we are not dependent on anybody for our requirement, and particularly for food grain requirement. But on contrary, there are countries which are thinking that if something can be produced elsewhere at much less cost and saving our natural resources, why should we produce such commodity? For example, China is a rice-eating country. China in past few years has reduced its area on rice cultivation from 32 million hectare to 28 million hectare. 4 million hectare they have reduced the area. The logic is that every kilogram of rice that we produce, we spend about 4,000, 3,000 liters of water. Why we should spend so much when we can get cheaper rice from other countries? When India can produce rice for us, why we should produce at the environmental cost? We feel proud in exporting 18 million tons of rice this year at a price of 65,000 crores, ever highest. But at the same time, you look at the cost that is put on the natural resources that we own, and that our future generation will ask for what kind of land, water, and planet we will be passing on to the next generation. So this is a real matter of concern where we have to really be worried about. The partnership, this is extremely important between countries, between continents. Why? Because, you need to have a comprehensive surveillance strategies for plant diseases that include strategic partnership among research universities, development agencies, non-governmental organizations, the private sector, and the CG Center. And one classical example of this partnership, which resulted in a very effective curtailment of the diseases, is in case of wheat blast we have seen. UG99 we have seen how it was identified on time, and then the partnership through CG system, through CIMIT, sending the material outside, whether it has to go to Bangladesh for blast screening or hotspot in Kenya for this UG99 race where it existed. This resulted in deployment of new genes through breeding into our upcoming varieties, and most of our varieties, they carry resistance to UG99 race and also to wheat blast. And this is one important example how partnership has helped in providing solution. You look at the donors that we are using in many breeding programs. In case of rice, we have the bacterial blight resistance. Dr. Mandal is here, and he would vouch for it that the isogenic lines developed in the background of IR24, they have become the principal source of resistance to bacterial blight in many nations' breeding program, including India. All the varieties that we have released in India, they carry either XA13, XA21, XA38, and XA13, 21, the genes, they come through partnership approach. That has expedited and led to release of large number of rice varieties in India. Almost 43 varieties through molecular breeding have been developed, and many of these are carrying resistance to bacterial blight and blast disease. If partnership was not there, if these genes were not identified, if they were not transferred in good agronomic background, if they were not readily available for our breeding program, we would not have been able to make such a contrasting, distinct, and disruptive progress. So partnership, again, is very important, and this kind of symposium that you are concluding is extremely important in finding partnership within country, outside country, and taking it forward for betterment. Then the surveillance and epidemiology. This research program focuses on disease surveillance and epidemiology for high impact plant pathogens and on major food crops such as wheat, potato, cassava, banana, corn, rice, are needed to prevent plant pathogen spread and predict the global burden of plant diseases. Currently, if you see in terms of disease surveillance, this is awfully underfunded, and there is no, this is not only on a global scale, it is done only for few crops like rust wheat in case of wheat for rust, and late blight in case of potato. See, for example, how much systematic surveillance program we have in India. As a, for example, you look at the weather forecast. For weather forecast, you have got a Mausam Vibhag with huge number of scientists, infrastructure, everything provided in place. But for studying surveillance mechanism, occurrence of diseases and pests in different areas, their documentation, strategies development, this is not a national priority. We may be doing here and there some studies at individual level. Even on our research program, surveillance is not a core research area. Now, this is required to be done. So this is one area where I think we need to do a lot if you want to control the plant diseases. The modeling, surveillance, and bioinformatics tools in many cases are ready to be deployed on larger scale and larger landscape, and this requires the research, phytosanitary, regulatory, and developmental agencies. But organized strategy is required for this purpose. New outbreak can be predicted with the disease surveillance system, and data can be used to determine the origin of outbreak, strains, and calculate the rate of spread. So this is important. Only if we do in a systematic manner, it will be possible. The global hotspot for new emerging diseases can be identified, and this is possible again with a systematic surveillance studies. The information will also be useful to deploy resistance germplasm, appropriate fungicide, or other eradication method at the landscape level. And there, I think a kind of community approach would be required in order to make effective control. For example, you have seen in past, not in terms of plant diseases, but in terms of insect, fall armyworm. Now, fall armyworm on this, our IRI farm itself, last year it came. It was present in everybody's kitchen garden. It was present in farm areas. Now in farm also, we are doing fragmented, research in different areas. There is some corn in seed production units, some in soil science, some in, DMR, and then some in IRI main. If you don't do coordination within institute, it's a small area I'm talking about, then many times your complete crop can be destroyed and you will not be able to, curtail it. So effective coordination among different groups. And that's what I said at that point of time, that wherever even a single part of maize is grown, people must ensure that they are applying Coragen or other control measures to control it. Otherwise, it will become a major problem. So this is high time, therefore, for strategic thinking, as the food security of resource-poor countries is threatened by both plant diseases and also COVID-19 pandemic. That has impacted food production, access, supply chain, and health of agricultural workers. Imagine if we did not have so much of food grains in our godown, more than 40 million tons of rice and wheat in reserve. The government has been able to provide 80 crores people for last 9 months free ration of 35 kg per month. Now, this was not possible if we didn't have enough production. And this is therefore, protecting our crops from diseases pest. This year, I have experience of rice crop traveling with the farmers. There are massive incidents of neck blast, also bacterial blight, and brown plant hopper, all three. And some cases, the farmers, because of neck blast and because of bacterial blight, they did not harvest the crop because the amount of labor required was huge, and they went for burning the crop. So this is, effective management where using precision agricultural technology, forecasting system. If we have a large area of suppose 100 acres, and if we are able to identify that there is a blast disease incidence has happened in one pocket, that selectively that pocket can be spread, and therefore spread of disease in the rest of the field can be checked. And this is where cooperative approach and farming using drone, which have sensor, biosensor, which can sense or take pictures if there is a foliar damage because of blast, and immediately you go for localized spray. And once the localized spray is done, then your cost of pesticide becomes extremely low. It will become 1/20th, 1/30th, and you don't allow the disease to spread. A very interesting observation I found this time when I went to farmers field in Karnal area in Urlana village. We have Dr. Saharan is a team member of our MGMG, and a farmer observation I found. It was so intriguing that in his field of Pusa Basmati 1401, there was incidence of bacterial blight. And when I looked at the spread of bacterial blight, there was a path of almost 2 meters running in length of more than 200 meters across the field. And this, generally we get, these kinds of harbor burn, bacterial blight burn in a specific area, patches. But this almost like a road, it was clean, in that it was damaged by bacterial blight. So I asked farmer, how come this kind of spread has happened? And then he said, sir, this is the blue bulls, the Nilgai Ghalroj, which has moved on this path.
Wow.
And while moving across the path, the animal is carrying the infection of a spore, causing damage to leaf while running through the field, and then injecting the spore through body into the leaf damage. And you get a complete streak of bacterial blight in the entire field. Now, those are the kinds of situation observations many times farmers tell, which is really interesting to record that how diseases can, bacterial blight, and how a blue bull can act as a what is called vector for bacterial blight. I don't know if Dr. Mandal would agree with that observation or not, but this is what I saw exactly this way the damage had happened. And wherever the blue bull had taken circle, it was burnt in the circular farm also.
Oh,
so that was, kind of good observation, and so we have to be watchful of this. I am sure that this symposium, the deliberation would have certainly considered some of these points to ensure better plant health and through plant health, a better animal health, human health, and this is what is called One Health program and food security of the nation. So I will look forward for the specific recommendation of the symposium, which can provide a roadmap as to in what direction we should move in future in terms of managing the emerging plant diseases in particular and diseases in general, so that the losses that happens because of plant diseases can be prevented. I'd like to thank you, each one of you, Dr. Rashmi in particular. I can see Dr. Subhas also, Dr. Subhas Chandra, Director NCIBM, for your patient hearing in this valedictory function. Thank you very much for inviting. Thanks once again. Thank you.
Thank you so much, sir. Very nice point, sir. We have noted down. Definitely. Survey and surveillance really is very important, sir. One time we really had a very strong program of Department of Plant Protection, Quarantine and Storage survey and surveillance program, but that now has fizzled out. But definitely all your points have been well taken, and we would like to bring good recommendations, and we'll give it to through society to ICR. Thank you so much, sir.
Thank you, sir, for your valuable message. So now I welcome Dr. Subhas Chandra, Director in CIPM. Sir, welcome to the Delhi Zone Virtual Symposium.
Thank you. Thank you. Namaskar, sir.
So Dr. Rasmi, I can also see Dr. Madhvan Gopal. Thank you, Raksa. Welcome.
Dr. Madhvan, these all are our very
Wonderful.
distinguished members of Indian Phytopathological Society.
Great.
Yes.
Welcome, sir.
Good to see you all.
Yeah. Nice.
So I have that, sports program of the students.
Yes, sir.
They are waiting
So quickly,
for
vote of thanks.
Yeah.
Kindly propose. a vote of
Sure.
thanks.
Very good afternoon to all of you. It gives me an immense pleasure to deliver the vote of thanks for this event. First, on the behalf of the organizing committee and the Division of Plant Pathology, I would like to propose hearty vote of thanks to our chief guest, Dr. A.K. Singh, Vice Chancellor and Director, ICAR IRI New Delhi, for kindly accepting our invitation. Thank you, sir. Your presence, sir, is indeed a source of great motivation. I sincerely thank our beloved chairperson, Dr. Rashmi Agarwal, Dean and Joint Director Education and Head Division of Plant Pathology IRI, who has been a great support behind this event. I would like to thank President IPS, Dr. Pratibha Sharma, and Secretary IPS, Dr. Robin Gogoi, for their constant help and guidance to shape the conference. I am very much thankful to all the speakers, reviewers, and technical committee members for their active participation and support to make this symposium successful. I want to express my gratitude to all the faculties and staff students of the division, all the participants, and all the IPS members from Delhi zone and from other zones also. A special thanks to Dr. T.K. Bag, President IPS Delhi zone and organizing secretary for organizing this symposium. My heartfelt thanks to IPS Secretariat, who all along supported to organize this function. So before concluding the program of IPS Delhi Zone Chapter 2021, once again, I thank wholeheartedly to everyone whose contribution has made this symposium successful. Thank you.
Thank you, Amrita.
Thank you.
Thank you, sir. Thank you, Dr. Madhuban.
Thank you.
Nice
Thank
to
you
see
very much. Thank you so much.
Thank you,
Thank you,
sir.
everybody.
Yes, seeing you after a very long time. So
Yes.
today I will be just going towards your side only, Dr. Madhuban. Anyway, thank you all of you and congratulations to all those who have got awards.
Yeah.
Dr. Bagh had to leave quickly, has he left?
Thank you.
He had a
Chalo, congratulations from my side also, Kuleshwar and all the
Kuleshwar, yes,
and all the
Shravya.
students and
Yeah, there were few names which were familiar.
posters
Thank you, sir.
Posters.
Thank you, ma'am.
So some of the
Thank you,
posters
everyone.
were really very nicely presented.
and
Yeah.
Kuleshwar, I
Right.
missed your presentation, but
Yes, ma'am.
there were other things going on, but definitely. And all the lectures
ma'am,
were,
for
Celia's was very informative,
mm,
and especially for all the youngsters. This survey surveillance program was at one point of time, it was very strong program.
Yeah.
And at that time, I had just joined the department in 1980-82, and it was constant. Dr. Saharan may be knowing, but he also joined late. But from IAWBR, also
Yeah.
initially DWR, we had a very strong program, and that was with IRI. When Dr. Joshi was at that time head, and he was a PI of the project. There we used to, like, I was not involved directly, but I knew that how strong that program was. And because of that only, we could identify the race, then gene deployment strategy, the Puccinia path. It was only because of that. And that only now also they follow the same, Dr. Sarah, you will agree with me,
Yes.
that if you are seeing any single pustule on wheat, maybe any rust, at that time stem rust was the predominant and then became the leaf rust, and now we have yellow rust. So now also the same strategy is there, that you in 1 meter square area, if you are finding 1 or 2 pustules only, you immediately convey to the farmers to spray. And that was possible only due to constant vigilance, surveillance. And but now other technologies are there, drone technologies and multispectral imaging. And then that also is same only. Through other latest technologies, one can spot the places where even single pustule is appearing, and then through drones also one can do that. But still, from theory to practical, little time is required for that. So
Yes, ma'am.
parallelly, as sir was also telling about the blast, neck blast and others, and for rice. So if that program would have been very strong, in rice, All India coordinated program, our wheat survey was also through All India coordinated program only. And for rice also, they may be there. Vishnu may be
But
knowing.
ma'am, wheat it is very strong
Very
with
strong,
all
yes.
the equipments and all.
Ah.
And rice, it depends, ma'am, depending
Ah,
on the.
the similar program, if rice also has a very
Yes.
strong throughout the crop season, and for especially hotspot areas from where it spreads. So that then really can be a very big input from that team, the survey surveillance. And Dr. Subhash also will agree to me that sir said that BPH this time also it was in a like major incidents was there for
Yes, madam.
BPH also, like neck blast and BPH and bacterial leaf blight also. So definitely,
Madam,
and
in this,
also the what Dr. Celia also said, no, for biosecurity of the country. Again, if survey surveillance program is very strong, then one can quickly detect the any exotic pests or pathogen coming or establishing in some areas. So that's a really very and baseline information you will get. That's the first-hand baseline information, real field information. Only thing is that some strong program with the funding. There were 2 jeeps at that time available with our division, and always they were on roving surveys. So that was funded by the Government of India, Department of Plant Protection, Quarantine and Storage. So similar program or otherwise with the novel technologies, one can always have now fresh programs. So Amrita, it's a good job for you also. Some points I have noted down. You, whatever sir has mentioned, they were very important points.
Yes,
like partnership, international and national partnership, and then survey surveillance program.
Yes.
And then he said post harvest and curtailing the losses, and then of course precision agriculture and how we can really say, like he said, no, that's nilgai or that bull which passes through the field and spreads. So like that, there are even humans can also, like sometimes, especially for the rust, we spores attached to our clothes and body, and it can give like travel to other places, and then you can spread such pathogens. So it's a very important session, and we are very happy that our director agreed and he gave us a lot of points.
Yes,
Ma'am, Subhash sir wanted to say something.
Yes, Dr. Subhash, please. Yes, I could see.
Madam, that you mentioned about BPH. No, BPH we have been observing for a number of years now, that first outbreak in 2000, and we have found very good correlation with the number of rainy days.
Yes, I know.
And that more than 30 rainy days during June to September. So that is means that is a very good indication of likely scenario. So this time also rains were good, and we found that is more than again this time that BPH outbreak was there.
Okay, so forewarning can be done based on that.
Yeah, because if by looking at the projected trend of rainfall, like weather forecast, we can have a very good indication where what will be the likely scenario, and then the timely preparedness of farmers.
Yes, sure.
So that way, I think long-term data, if we have, we can analyze.
You can analyze and come out with an app.
Very good clues, no? Very good thumb rules. Yes.
Like we have in Rust also.
Yes. So only thing, long-term data and keen observation, I think that is the key for the survey surveillance.
Yes, nice to see you, Dr. Madhuban. You want to say something, Dr. Madhuban Gopal?
Ma'am, what I understand that today's discussion that we should resume again a strong group of monitoring, at least from plant pathology and in collaboration with Dr. Subhash Institute. I think that will help us also, because already we have an independent program in AICRP. Right, we have to report the new emerging diseases, other things. But I think from our point of view, at least from the existence of these divisions point of view, we should have a strong group. I think you can remember that we had earlier strong group of means at least individual scientists of different major discipline, sub-discipline. So that way we can constitute. So this.
Yes, definitely. I fully agree. We have one vehicle with us. At least we can utilize that, and we can join with the NCIPM. They also have a mandate. I think NCIPM has a mandate.
Yes, madam. Yes.
So our division scientists associated with different crops, they can join hands, and we will have a common program of survey and surveillance.
At least for the major crops like rice,
major crops,
maize, all these.
Maize, wheat, and
Sure,
some of
madam.
the emerging diseases are
Yeah,
coming in these crops. Definitely, like we have now Fusarium head blight. You know,
Yeah.
earlier it was only for cold loving places, only temperate regions. Now we are having in tropical, subtropical regions, the Fusarium head blight. And high temperature loving species have predominantly now affecting the wheat crop. And similar in many of the yellow rust races, which are warm temperature loving races. So such programs can always be. I remember when Dr. Subhash, when Chirantan was director,
Man.
he also at that time wanted to have some collaborative survey program.
Yes.
So, but
For your information, madam, this time we have one with plant pathology, virology, one project.
Yes,
we have
yes.
formulated
Yes.
one for
Yes.
cotton. In that,
Okay, got it.
what is that
And
leaf blight
one
in the
mission project, Dr. Baranwal, if you recall, you know, from ICR, one platform project also has come for some of the emerging diseases.
That
with
is
NCIPM.
yet to come. It is
Yet to
proposed
come.
at
Yeah,
the
it is proposed,
yes.
but so under
Yes.
that, I think some of these
For
objectives can
emerging,
be,
yes.
emerging diseases.
That new
Yes.
EFC, that program is in
Yes,
Yes.
New EFC.
EFC not yet like finally
Yes,
approved, but it's a proposed project.
Can I make one suggestion?
Yes, sure, Dr.
Yeah. We have been talking about surveillance, but a definite program. It is not that any other day we are going somewhere, getting some data and coming back.
No, it's a regular.
So we have to really design in a very systematic manner that according to the growth stage of the crop in a particular area, one has to go and collect the samples systematically, not randomly. Some statistical methodology has to be adopted, either stratified sampling method or whatever it is. So only then some scientific output will be there. Otherwise, it will be just reporting this much incidence, that much incidence.
That only individual scientists, as they are going now also, that's a different work altogether. But if some constant vigil, survey and surveillance means a constant program with all proper procedures and following statistical sampling procedures. That was there earlier. So now also if some scientists come forward and that can be decided and a proper program can be framed, at least for some of the important crops.
Yes.
There some emerging diseases are coming. We cannot have a very elaborate program for all kinds of crops and diseases. But
like NCIPM, if they have started for cotton, we can join hands with them for cotton also.
Madam, in this actually, Dr. Varnwal said it is very, very important. Like when we go to field, so that optimum sample size, that is very, very important.
Yes.
Because sometimes if we observe on 2 plants, 4 plants, 5 plants, or 10 plants, it will give us some output. But whether that is really reflective of the real situation in the field, so for that robust statistical mechanism is very, very important.
Yes.
So that optimum sample size depending upon means the population or intensity or that severity, that is when sometimes we ignore that because means very tentatively we observe on some plants and report that. So that proper statistical procedure is very, very important.
Yes, Dr. Subhash, I agree. And whole crop data has to be there, the variety, the location, the environmental factors. There are many other factors which performa is already existing. One can always devise those performa, and now we have GPS system also, so the location and other things will automatically come from where you are sampling. But definitely the statistical procedures have to be followed for sampling and recording the disease. Yes. We can now.
Yes.
Dr. Gogoi, yes.
So I would like to congratulate Dr. T.K. Bag and Amrita for successfully completing this function. So they came forward to take responsibility of for 1 year, and even though they tried for organizing in physical mode, but could not happen. And also I congratulate all the winners of different poster presentation, particularly those students, and also Kuleshwar and Firuz Mandal. I welcome both of you for final contest. So that will be held in the month of March. This is 23rd to 26th March 2022 at Jaipur. This is Javner Jaipur. And also, I invite all of you who are present now, so to join after 1 hour. And this IPS talk series lecture, very important. Just now, our head madam, Rasmi madam, mentioned that how rust also can cause infection in the human being. The topic of this today's lecture is crossing the kingdom barrier, human diseases caused by the plant pathogens. That will be delivered by Dr. G. Ravikant. He's from Bangalore. So the talk will be started at 4:30. I hope all of you will be joining and trying to get the information how this plant pathogen can be the human pathogens itself. As we know, this after COVID.
From kingdom. It's a transfer from one kingdom to another kingdom.
Right.
Yeah, they are there. They are all quite fast. Yeah.
Like black fungus. After COVID only, we came to know in a wide range how the impact of black fungus.
Black fungus, white fungus, candidates.
Yes. So from my side, thank you, Amrita and Dr. Bag. And also, I thank all my students and the faculty, these members, and also from this NCIPM and NPPGR.
NPPGR. Yes.
Yeah, because always we are working together. These three institutions from PUSA campus. So
thank you. From my side also, congratulations to Bag and Dr. Bag and Amrita.
Thank you,
and all others. Thank you, Dr. Bikash, Dr. Kalyan, all joined together and made it a success. Dr. Gogoi and your office. Thank you.
Robin, one more point. In future, let this event be for 2 days. They should not go back to the original half a day or 1 day.
Once
actually, we proposed for 2 days because 1 day is very hectic. So some last time what we found that some could not able to listen the Narasimhan Award, and side by side the poster session also going on. So that's why we thought that we should separate these two things in 2 days. That's why.
And participation should be more.
Yes.
Yeah, that is very bad actually. Less than 50 only.
I was very hard.
1 day it crossed little over.
I was feeling that your director has joined and not much participation.
Number is very less.
Yeah. You can send the link across the zone.
Across the zone,
yeah. At least North Zone or some people who really. And we have invited key speakers from that zone, so we should have other zone people also. But listeners can be from any zone.
Madam, actually the link was sent to every zone also. Means, this symposium.
Normally, people are busy and there are many other things going on. So
that means other zone nobody has participated. It looks like.
It looks like.
It's only 20.
Today, because of e-poster presentation and students' participation, the number in morning was good. Because they all had to present, so they were listening to each other's presentation also.
Yes, madam.
You can send out the message to zonal presidents and
zonal president. You can even from secretary side, some message should go, because we are calling such senior dignitaries, and then we see that the participation is less. We don't feel very good and comfortable.
It's only 28. That's not
now,
a good number.
Now they have reduced, but it was around 40.
Around 42 was there.
42
40 to 44 was there.
I think the message was very short time, and you could have a good gathering. It would have planned very earlier. So because virtual, we should take the advantage of this virtual mode of involving others zone also. So that we could not make it because maybe we have shortage of time. So that
started late maybe.
Yeah. Okay, so that we have to take care in the next year onwards.
Okay. So thank you. We'll wind up now. Thank you, Dr. Subhash and Dr. Celia.
Dr. Celia and
Dr. Subhash from other institutes.
Thank you,
Dr.
madam.
Jameel Akhtar.
Thank you, Doctor.
Dr. Vishal Somanshi from Nematology.
Yes.
Of course, our own division scientist, Vishal is. Yes, Vishal. Okay.
Okay.
Bye-bye.
Thank you, Amrita. So can we leave? Because
yes, you have to give us the permission to leave.
Yeah.
Okay.
Definitely. Thank you. Thank you, everybody.
Thank you. Yeah.
Without any cup of tea.